Thank you for those kind words. Good morning to all of you, and good afternoon and good evening for those of you who are joining us online. This is amazing that with these new tools that we have available, we're able to connect and participate and listen to what's happening here. So it is a privilege. It's also a privilege for me to be able to share and talk today on this topic following two amazing lectures by Dr. Bonagora, who talked about the whole clinical aspect of the disease, and Dr. Laflamme, who touched upon various nutritional approaches that could be taken. The approach or the talk that I'm going to give today is going to take a slightly different tack. I'm going to use our study with Myxomatous mitral valve disease as a case study to be able to illustrate a systems biology approach in trying to unlock nutritional solutions that can make an impact for the pets and the people who love them. So let me begin with a quick outline of what I'm going to cover today. I want to touch upon and go through in a little bit of detail on what I mean by the systems biology approach, giving you an overview. The other advantage of being the speaker on a second day is yesterday, several speakers touched on this topic. We heard a lot about omics and how that's used. So this will make it easier for us to go through that. Then I'll use the study on MMBD as a case study to be able to illustrate how systems biology tools can be applied to be able to understand pathologies or perhaps changes that could be addressed through nutrition. And then finish up with some early microbiome studies that were also conducted by Dr. Johnny Lee and team, also in relation to MMBD. So what is systems biology? It's a name, it's a term that's used so often. But in a nutshell, it really explains biology in multiple interactions. And this is where we want to step back and take a look at all the amazing advances that we have made in biology and other fields over the years by taking essentially a reductionist approach. We try to take relationships on changing one thing on something else, and it has really opened up the field and given us a lot of insight. But as you look around, we quickly realize that just a reductionist approach is not the ideal way to do that, because in biology, you have complex interactions that play, and the outcome is decided by not just one thing, but by many things. And what we do with systems biology is to be able to take into account all those varied interactions and try to get an understanding of how that influences the biological outcomes that we're interested in. And this is a science and a field or an approach that's happening in a lot of other fields. Engineering, Google Maps, for example. I was driving in from the airport to the venue, and when I looked up my timing of arrival, after a few minutes, it quickly doubled because they factored in the traffic that comes in. So the other thing to keep in mind is that you're not only looking at multiple factors coming in, but how can you make that analysis much more dynamic and take into account that there are things that are happening in biology that could, again, influence an outcome or a physiology that you're interested in. We're not quite there, but again, with some of the tools that we have available, these things are becoming a reality. And for people like me, researchers like me, this is an amazing time to be able to do science, because you have the analytical tools and the mathematical muscle, so to speak, to be able to ask and answer these fundamental questions. And again, just to illustrate simple relationship between enzyme and a substrate, old studies that many of you grew up when we started thinking about classical biology and genetics, and now we've gone into regulatory networks of genes. Now, one important thing that we need to understand and keep in mind is that when you're talking about complex interactions, you also need to take into account the ability to be able to generate complex data sets from multiple components. And now again, with some of the technological advances that have happened, we are at a better place to do that, and more is yet to come. And combined with just the wet lab tools and the omics tools that I'll talk about in the next slide, you need to have significant computational power to be able to combine all that and paint a picture, so to speak, of what's going on at a given time in a dog or a cat or a human or any biological system. And the most important piece for that is you need to have constant interaction, discussion, collaboration with the biologists to be able to make sense of all that. And again, historically or traditionally, many of us grew up with mathematics being more engineering focused rather than biology, and many of you here perhaps have had that approach when we went through our education. But what's emerging now is that those two fields are coming together much more closely, and that is where you're able to unlock the power of these tools. Because having a large data set without the analytical ability to be able to analyze, interpret, and see patterns, and then bring in people who can take a look at the data and make sense, saying this is what this means biologically, clinically, is critical. And ultimately, the whole idea here is to be able to take a reductionist approach when you look at all these different sources of data, where you're trying to ask the question, what's happening to this particular gene, but with a set of so many genes in play, and then bring it back up in a holistic manner, because ultimately what we need to do is how can we impact the health and the life of a particular pet or a human. Without that, this just becomes a scientific pursuit which doesn't have a clear end and goal. At least that's how we view it. And we'll talk about some of the examples as we go through the slides. Now, this is a description of the tools that we use for systems biology. And what you're seeing here is the classical pathway that all of us grow up with: genes getting transcribed to mRNA, getting translated into proteins, proteins breaking down various things in the body, resulting in metabolites. And to just give you an illustration of the amount of genes that you're talking about, in a dog, you're roughly looking at about 20,000 genes, translated to about 40,000 transcripts, and that eventually gets into over 1 million different proteins that could be generated on that. And metabolomics perspective, there are over 3,000 to 4,000 metabolites that are being produced in an ongoing basis in a dog. In the cat, it's more or less the same. In the human, roughly the same. The gene numbers are significantly higher. Now, what you also need to keep in mind is in between these lines, there are other changes that we are becoming aware. A whole area called as methylated genomes or methylomics. How does these methylations impact the transcription and translation of these genes? Because you have 40,000 transcripts, many of those transcribed genes don't really end up as proteins. They also have regulatory function that we are really unraveling now, and there is a lot more to be able to come from that. What's amazing again is the technology that have come up to be able to ask these questions. So if you look at in the past, genetics was just trying to take a small piece of DNA and sequencing it. Today, you can do high throughput sequencing where you can essentially sequence all the milieu that's present in the sample, whether the gene or the DNA or the RNA comes from the host or something else. Same thing with proteomics. Instead of looking at single proteins using an ELISA or a similar Western blot type assays, you can now do sequencing across the entire spectrum of protein that you can find in a sample. The same with metabolites. Now, an important thing to keep in mind as you look through this picture is this is only telling half the story. Let me correct myself. It's probably telling maybe 20% or 10% of the story, because as you all know, we have so many more microbiota or bacteria, viruses, etc. in our body than our own cell types. And for each of those organisms, maybe they have only about 3,000 genes to begin with, but they have exactly the same patterns emerging. And as Dr. Sukudowski explained yesterday, it is clear that that is essentially an organ that we need to consider as we take a look at things from a systems biology perspective. So they have their own genes that get transcribed to proteins. They metabolize various things, and we saw examples of that yesterday, how they can influence health, disease, mood, behavior. And this is, again, as I said, we are sitting on an amazing amount of data that's going to emerge in the next few years that's going to really revolutionize how we look at biology and how we use this as tools to be able to make changes in diet, health, nutrition, or even disease and drugs. So how do we use systems biology to be able to ask the question that we are specifically interested in? And the goal here really is to be able to define targets. We covered all the tools that I went through. And what they will lead to is identifying targets that we can hopefully influence through nutrition. Now, all targets can be influenced, that's a given. But this gives us an ability to be able to very quickly understand the biology or the pathology that's happening in a particular system and ask the question, how can we influence that with the known nutritional solutions or approaches that we have? And this is, again, where you have to have a crosstalk between various groups, various experts collaborating to be able to bring all these things together. And then you use those, if you're lucky to be able to find a set of targets that you can influence through nutrition, you can then use that as a means to be able to either slow progression, and in some cases perhaps even prevent the disease. And that's really the holy grail of how this will all emerge in the future. So let's move on to the case study that I want to use to illustrate this approach. We have gone through extensive discussions about the disease per se, and Dr. Laflamme also covered quite extensively all the nutritional approaches that could be taken. So the focus here is to be able to see how we can use these tools that I described as part of systems biology to be able to identify targets that we can influence, come up with a testable hypothesis, and do some studies to be able to confirm whether you actually influence those pathways or not. This is something that we went through in detail, so I'm not going to belabor the point, but again, it's clearly a huge problem that needs addressing. There are some clear drugs available that Dr. Bonagora covered this morning. But it's a disease that's common in small breed and geriatric dogs, and the causes are unknown at the moment. So again, going back to the approach that we took was to characterize the underlying biology or pathology, hopefully define targets that leads to hypothesis generation, and then validate these hypotheses either through studies and in some cases even in vitro experiments, which is obviously not the case here, but that is again where this whole field is evolving. Let me make a comment on that, which I think is also a part of how we look at systems biology. It's not just the omics space, but there is significant development happening in cell culture models and organoids, which many of you might have heard about, where you're looking at cells taken from a dog or a cat or a human or a mouse that can then reorganize into forming tissues. They're not exactly the same as a living tissue in the animal, but are quite close in mimicking some of those characteristics. But you can again ask these questions in terms of how that particular tissue or organ will respond to an intervention. And again, we're not going to talk about this in this particular topic because that is not approach, but I just want to plant that seed in your mind as we think about the scope of this approach beyond just the omics. So I'll be covering two different analytical tools in this particular approach, metabolomics and then transcriptomics. This was a small study where we had 18 dogs with preclinical MMED, 11 control dogs. We did an untargeted metabolomics using the Metabolome platform. Over 506 metabolites were identified, and of those, 54 showed different levels between groups. And this has been published in the Omics Journal in 2015, and I have the reference for that as well, and you can get it from the QR code that was shown. For the tissue transcriptomics, Of course, a smaller set of dogs, because these were tissues that are obtained from these dogs. Mitral valve, we had 3 MMB dogs versus 3 control dogs, and we had tissues from the left ventricle, a smaller sample of 2 MMB versus 4 control. Over 25 million sequences per sample were generated using the Illumina platform. And then, of course, bioinformatics and statistical data analysis. And then we ultimately came down with differentially expressed transcripts, which are 812 for the left ventricular sample and 264 for the mitral valve samples. These are the papers that I was referring to. Again, these are some of the early papers, I believe some of the first papers in this field that Dr. Lee and his team have published, looking at untargeted metabolomics of serum samples, as well as RNA sequence transcriptomics of cardiac tissue. There was another larger study, which was much more dogs involved, 27 control dogs, 22 that were B1, 22 that were B2, and then dogs that had complete heart failure, and all these have been published. And the results are kind of a summation of all the work that these papers talk about. So let's look at what are the significant changes that were observed as part of this analysis. And I'm going to talk about the key ones that really make helping us to drive the hypothesis, but there are others that we did not get into, and we can have a discussion later, or the paper will give you more information about that. Now let me build this slide out. And we heard about from Dr. Laflamme's presentation that heart is an organ that prefers energy sources coming from fats. 70 to 90% of cardiac energy requirement comes from fatty acid oxidation. It's also a very highly energy hungry organ for obvious reasons that you all know. And when you start looking at the transcripts that were differentially expressed between healthy and dogs that had MMBD, they were clearly identified as certain proteins that are essential for fatty acid oxidation, that are proteins that are important to bring the fatty acid into the cell and then essentially initiate the oxidation process in the mitochondria. And the 3 that really stood out have been called out: fatty acid binding protein, fatty acid transporter, and then long-chain fatty acid CoA synthetase. The other important finding that we saw, as in addition to fatty acid transportation, all again related to energy metabolism, remodeling that have already happened in these tissues, was the increase in GLUT, which are the transporters for glucose, suggesting that the energy metabolism was already shifted from fatty acid oxidation to glucose oxidation. And then you also see an increase in both increase in circulating glucose as well as lactate, suggesting that the energy pathway is generating a lot of waste products that really suggest that there is a lot of anaerobic glycolysis happening as compared to fatty acid oxidation or just straight glycolysis. And these things are important because they have negative connotation in terms of the reactive free oxides that are released that can have damage, as Dr. Laflamme covered in her presentation. We also see signs of that when we start looking at increased oxidative stress in these dogs. And as you know, mitochondria is the energy source for the cell, and the amount of reactions that happen there to be able to generate ATPs creates a lot of free radicals that can get out and then cause damage. And as we learned from Dr. Laflamme's lecture, glutathione is a very powerful antioxidant, and it can neutralize reactive oxidant species. But as you look at the presence of these molecules in the samples from serum, what you can see is that there is clearly an increased sign of oxidative stress, because you can see an increase in the oxidized form of glutathione, which is a sign that you're not able to completely remove all the free radicals that are being released from these mitochondria, which are in excess, which are producing energy in these cases. So what you see clearly is a significant shift in energy metabolism, and as a consequence, you also see increased oxidative stress. Other markers for compromised fatty acid oxidation are acylcarnitines that are intermediates in the process through which the fatty acid oxidation takes place. And you see an increased accumulation of over 22 different acylcarnitine species in circulation in the dogs that had MVVD as compared to those that did not, and it correlated with the severity of disease. This is, again, metabolomics data, so we are combining both the omics, transcriptomics, and the metabolomics data together. The other point that was also observed as part of the metabolomic analysis was there was a significant methionine deficiency in MVD dogs. And you can look at the correlation as the severity of disease increases, you start seeing reduction in the methionine. And again, between the control group and the groups that were in type B disease, you see a statistically significant reduction. Now, why is methionine important? Methionine is important because it is an important precursor for the biosynthesis of carnitine, which, as we learned again from Dr. Laflamme's lecture, is an important molecule that's required for complete, efficient fatty acid oxidation. So again, as you put all this data together, you're starting to see a clearly emerging pattern of changes in energy metabolism, consequences in terms of some of the damage caused by free radicals where you see glutathione changes. So you're looking at both energy metabolism impairment as well as increased stress or oxidative stress in these dogs. So here's kind of the key takeaways from the studies that were done with the omics platforms. Compromised fatty acid oxidation and ATP production, altered amino acid metabolism that we saw, increased markers of inflammation and oxidative stress, reactive oxidation species getting out also results in certain level of inflammation, and we'll see some of that in the follow-up study that came out of this work. So this is where we then take all this data together and say, okay, how can we address that through nutrition? And here are some if we can statements that kind of help the team come up with a potential blend that could be used to address some of these challenges. So the question was, can we improve fatty acid oxidation? Perhaps providing an alternative energy source, provide antioxidants to be able to help cope with the oxidative stress that is likely running rampant in these dogs. Also try to control the inflammation that is also likely present. And then supplement some of the key amino acids, which in turn will help some of the pathways that are important to have an efficient fatty acid oxidation process. And the question then would be, does it give us an opportunity to be able to perhaps slow down the progression of the disease in these dogs? So now what we saw here was a whole bunch of omics data being generated from samples, serum, and in this case you saw tissue samples, putting all that together with analytical tools to lay out a picture of what's likely happening in the tissues of these dogs that have a disease. And that then helps you start looking at a way of generating a hypothesis and how can we then test it, looking at solutions that are available from our toolbox to be able to address. So that, I think, is the entire path of systems biology that I wanted to illustrate, and we'll go through how the study went. But again, just to step back and put this in perspective, this is an approach that I think is applicable for a lot of things that we're doing. And again, having these tools at our hand, the availability to be able to have samples that are appropriate, can really open up tremendous opportunity for us to be able to address a lot of different diseases or alterations that happen in dogs, cats, and obviously humans as well. And you heard from Dr. Jerome Fage yesterday in his wonderful presentation. for us to be able to use the approach across the board, across multiple species, both humans, dogs, and cats, which is unique in the kind of research setup that we have, we are really excited to be able to leverage these tools across platforms and see how these relationships can drive both solutions for us, and perhaps some of these learnings can also be translated back to humans. And this is also a point for us to think about the whole idea of One Health approach that we talk about a lot. But our solutions tend to be very targeted towards dog and cat. But with some of the work that we're doing with the larger NR group, there's an opportunity for us to be able to take some of these learnings across perhaps for some of the human challenges that people face as well. So, what was the blend that the team came up with? And again, here are the highlights. MCT, amino acid, omega-3 fatty acids, vitamin E, and magnesium. And I'm going to quickly stroll through this because Dr. Laflamme covered in detail the benefits of many of these things. You're talking about medium-chain fatty acids, C8 and C10, again as an alternative energy source for the cardiomyocytes. Lysine and methionine, essential amino acids for dogs. Methionine, we saw earlier, is important for carnitine, which is critical for the fatty acid oxidation pathway. Taurine, which we again saw from the earlier presentation, maintains heart contractility as well as is an antioxidant, again helping with some of the free radicals that are generated because of alteration in the energy metabolism. Omega-3 fatty acids, known as anti-inflammatory agents, antioxidants, can also have other benefits like lowering blood pressure, helping with heart rate and rhythm, as well as muscle loss and cachexia in dogs, which apparently is a big problem with some of these dogs getting into early or even late stage of disease. And then we did a small pilot study that fed dogs containing a diet containing this blend, and this was published in the BMC Veterinary Research Journal. And let me quickly give you an overview of the study, and I'm not going to get into the details of the study because this has been presented earlier in this forum by Dr. Johnny Lee. I'd like to just go back and loop back and say, here's what we started with regard to the systems biology approach in trying to unravel pathways that are not functional. And the key question, have we corrected some of those pathways in the analysis from the samples that came from these dogs? So this is a blinded, randomized, placebo-controlled study. 19 dogs with the MVD, 17 breeds, age 6 matched healthy control dogs. They were randomly divided into the two groups. They were fed their assigned diet for 6 months. And all the analysis for the echocardiography was done by a board-certified veterinary cardiologist at 0, 3, and 6 months. And this individual was completely blinded to the diet treatment. And then we looked at 6-month changes in LV and LA in both dietary groups. And here are the key summary results of the study. CPB, which stands for cardiac protection blend, reduced left atrial enlargement, ameliorates mitral regurgitation, slows down the progression of MVD in this study, and then it was safe because there were no other issues noted in the healthy control dogs. So having tested or validated the hypothesis with which we started the study, let's go back and try to look at, did we make corrections or changes in some of the pathways that really helped us drive to where we are with this particular blend. And this has been already published, so let me quickly go through. Let me go back. So what you're seeing again is a metabolic profile done as a PCA plot, and what you see is before and after picture. In the before picture, both the control and the healthy dogs, there seems to be, this is the baseline values, and you don't see much of a difference. But when you start looking at the 6-month data, you see a clear difference where you're seeing the dogs that were fed the CPB blend you have a complete shift or changes in the overall metabolic profile, which is what you would expect, and at least that was the hypothesis. And we dig down deeper and try to understand individual components that we changed. Here are some of the key features that we saw. The blend decreased the omega-6, omega-3 fatty acid ratio. We all know that omega-3 fatty acids are pro-inflammatory. Omega-3 tend to be anti-inflammatory because of the pathways they influence. And the ratio is critical to be able to establish whether you're decreasing the pro-inflammatory profile in these dogs. And that, as we saw earlier, is a risk for cardiovascular disease. And as you can see in the inset data there, you actually see an increase in the dogs that were fed the control diet, but in the dogs that were fed the test diet, you can see a statistically significant difference when you compare the 6-month data to the baseline, clearly showing that you are trending or reducing the inflammatory signature in the direction that you'd like this to be, which is lower inflammation. When you start looking at the usage of medium-chain fatty acids, caprate and caprolate are hydrolytic products that come out of MCT usage, and when you see increased levels of caprate and caprolate in circulation, you're talking about increased bioavailability, and that the system is actually using these alternate energy substrates. And as you can see in the inset data there, if you look at C10, you see an increased level of caprate in the dogs that were fed the CPB blend as compared to the control dog. You see the same also in the C8 data, clearly showing that the dogs are now capable of metabolizing this fatty acid, and you have actually influenced or shifted their energy metabolism back to the MCT rather than the other pathways that they were remodeled to use in the past. You can also see data suggesting improved fatty acid oxidation. Now, increases in acyl carnitine signifies incomplete fatty acid oxidation, which is one of the key findings that came out of all the systems biology and the omics studies that were done earlier. And when you see, what you see here is that dogs that were fed the CPP blend had significant decrease in 3 acyl carnitines that are listed out here. And there was also an increase in carnitine precursor. And as you can see in the data, as compared to the control-fed dogs, those dogs that was fed the CPB blend, you have reductions that you would expect in the right direction, clearly showing that you not only shifted, but you likely corrected some of the fatty acid oxidation issues that these dogs suffered from. So again, circling back from where we started, which is identifying pathways that have gone awry in these dogs that likely are the cause of the disease, you come up with an approach to be able to identify what those are, come up with a hypothesis of how you can influence it with the nutritional toolkit, so to speak, that you have, do a study where you're able to show some changes in the clinical conditions. But when you start looking at the metabolomic data, you're coming full circle back and saying you have actually made some of those changes that you hypothesized and started the whole study with. So CPP blend changes the metabolomic profile, suggesting that improved fatty acid oxidation, which is the second bullet point. You also reduce the inflammatory response, which is believed to be one of the drivers or one of the factors that influence disease in these dogs. And the alternative substrate energy substrate in circulation again shows that shifting them to an MCT diet approach is correcting some of the metabolic pathways that were wrong to begin with. So again, illustrating the power of the approach, using this as a case study to see how we are able to start with a hypothesis, test it, and reconfirm it. Let me move on to the next part of the talk, the last point, where I wanted to cover some of the gut microbiome studies that were done part of this project as well. This has been already published, and I'll have the publication here. But gut microbiome, as we talked earlier, is something that is becoming more and more recognized as a huge contributor both for our health and disease. And in reality, we don't know a lot about that. Many of the information that we have in the microbiome space, as applied to dogs and cats, we really import or learn or translate from what's known. Again, known quotes, because again, lots not known in the human space as well. So there's more studies that needs to be done in this area. But there is a clear correlation with changes in gut microflora and how it influences health. And here's an example of that with the MMBD. So for this study, 92 dogs, samples were collected. We had 17 control dogs, 17 dogs that had B1 disease, 23 dogs that had B1 disease, 27 dogs that had B2, and then 25 that were in the advanced stages C and D. We did our 16S ribosomal sequencing to identify the bacteria that's present. We also used the PCR-based fecal dysbiosis index that Dr. Sokurowski talked about extensively yesterday. And then we measured fecal bile acids in the serum using the Metabolome platform. And again, the correlation that we like to draw here is how does changes in microflora in the gut correlate with severity of disease? And is that a relationship to some of the metabolites that are being circulating in these dogs, most likely coming from the gut microflora? So here are some of the summary results from this analysis. So there are two pieces of data that's shown here. One is a diversity index, and the other one is dysbiosis. And again, as you all know, when a microenvironment or an ecosystem starts losing diversity, it's usually a sign of disease that ensues fairly quickly. So what you are measuring here is the lack of diversity that is clearly apparent as dogs get more and more into severe disease. That is clearly shown here. Control is A, and then you have the other three groups. It also correlates inversely with the increase in dysbiosis, and this is data from the methodology that Dr. Sokurowski explained yesterday. What you can see is that as dogs get into more severe disease, you start seeing an increase in gut microflora dysbiosis. Again, correlating with what's generally known in this area, and again, there is other studies that are being looked at in the human space where there is relationship with gut microflora changes and heart failure. Now, when you take the microbiome data and overlay it and see what do you see in terms of metabolic changes in the serum of these dogs, you can clearly see that trimethylamine N-oxide, TMO, which is a uremic toxin whose biosynthesis specifically happens because of gut microflora, they are significantly increased in the dogs as their disease becomes more and more severe. That's the top graph there. You also see abundance of E. coli, which are now believed to be having deleterious effects, health effects, also increasing quite significantly, if you see the lower graph, in the dogs that as they go through and get into more severe stages of this disease. Now, there is data and other studies that have shown that the E. coli genome is encoded with genes that can actually drive the synthesis of TMAO, and therefore there is a correlation with changing microflora, with the dysbiosis that we see earlier, and then increased in a uremic toxin that is critical. The one piece of data that was also there that I did not include was Clostridium hieronymus, which Dr. Sokurowski talked about yesterday. Again, you start seeing patterns which really suggest that what he was talking about yesterday, where changes in those also reflect severity of disease. So let me summarize quickly the results of the microbiome study. And again, this is just an early study. There's a lot more that needs to be done in this space because, again, from a nutritional standpoint, the belief is that we can influence microbiome to a certain extent, either through prebiotics, fibers, perhaps even probiotics. The jury is still out there. So this is, again, another tool that we can use to be able to address challenges in this disease and others. So what you can clearly see as a summary of the results from the study is that gut dysbiosis is clearly associated with severity of disease. As the disease becomes more and more severe, the dysbiosis index goes up, the diversity goes down. It's possible that gut biosis starts at an early preclinical stage before heart failure. Again, this is just a hypothesis at this stage, something that needs to be proved out. And then a key metabolite that has got a bad rap in many circles, including in the renal space, which is TMO, is associated with increasing progression of MMBD. And we believe E. coli, where we also saw those numbers go up, could be the likely source of how this particular compound is being generated. And then increase in C. hieronymus is also linked to MMBD because that's an important bacteria that converts primary bile acids into secondary bile acids. Again, more studies are warranted in this space. Now, let me again step back and bring up one other point that is also important as we talk about the systems biology approach. The ideal goal for us as we start looking at complex diseases, nutritional challenges, is to be able to intervene as early as we possibly can to be able to influence the outcome of the disease. That's at least the approach that we would like to take. And in many of these diseases, before you start seeing overt clinical signs, the train, so to speak, has already left the station. So how do you start looking at a way to be able to address some of these things earlier? And this is where the idea of biomarkers become more and more important. And that is one aspect of systems biology which I think is important to highlight because some of these tools also help us identify markers that you could potentially use earlier in the process to be able to identify changes that ultimately will lead to disease. In this particular case, before heart remodeling happens and the disease progresses down the path, can we come up with a solution earlier? And many of the tools that we're talking about here are really ideal to be able to also start defining biomarkers. Because the pathways that are changing in this and other diseases indicate to something that's going down if you have a solid hypothesis and have validated it. But the biomarker conversation also becomes a little challenging because biomarkers are indications of what happens down the road. They're not diagnostic tools, and there is always that debate, and that's something that I'd love to have a discussion in the session later, because you have to look at it both from biomarkers that predict biology going down the road, eventually leading to disease, versus can you start looking at it as a pure diagnostic tool? And that's the debate is happening quite actively, and hopefully there'll be changes emerging because as we start getting more and more data from some of these tools, it's going to be apparent that these things are going to be important in helping us make decisions for your patients, for human patients, with regard to solutions that you can offer to them to address these changes. Let me finish up with a slide that I wanted to highlight, because this is ultimately the approach that we're trying to talk about here and that we'll leverage quite extensively in other things that we're doing. It starts with a problem that you've identified, that you want to look for a solution. And using all these omics tools, you start defining that in terms of pathways, in terms of areas that you can influence using nutrition. You do experimental analysis, data analysis, have hypothesis-driven models. And this idea of dry experiment is important because as some of these tools become much more powerful, we have a lot more data, we have a lot more understanding of what's normal versus not normal. And as you know, there's a lot in the human space, a little less in the companion animal space, but even in the human space, there's a lot that needs to change. But once you have those defined entities of what's normal, you can start doing experiments, not necessarily by actually doing feeding studies, but simulations of what could happen. And it's interesting that Dr. Bonavera mentioned AI. Those are tools that are being now used to be able to look at echocardiograms, for example. We can leverage the same kind of tools to be able to look at the data set and say, okay, what are things, if you change these things, how does these pathways shift over time? And that'll help us perhaps narrow down in terms of areas that we want to influence, and then come up with solutions. And ultimately, you have to do the experiment with the dog or the cat or the human to be able to demonstrate that. But this really helps us focus and identify earlier things that could be addressed, do some preliminary experiments either in silico, in cell culture models that I talked about, before doing the actual experiment with the species in mind. And part of that is going to be biomarkers. What we learn from that could essentially be pieces of information that you can help drive decision-making down the road in terms of how you want to influence and what kind of solutions, options that you want to offer up to your patient to address a particular issue that comes up. Thank you for your attention.